so much. So first and foremost, in the back, can you hear me? Thumbs up? Okay, awesome. So I just have to say, first off, thank you, Lisa, and to the other organizers of this series for giving me this opportunity. I am genuinely excited to be here and speaking to this crowd. I'm looking forward to hearing what you think about what I present, your thoughts, and your questions, and sort of exploring this with you. So what I'll try to do is, as I go through this work, because we're going to sort of cover a wide span of different topics related to microbes. I'll try to take some pauses and maybe get some questions in the middle, but I'm hoping overall we'll have plenty of time, and I'll definitely be here at the end as well, after 7 if you want to chat some more. Okay, so first off, who thinks microbes matter? I'm done. All right. No, that's awesome. Okay, so easy crowd, cool. So I think microbes matter a lot. I love them. I love bacteria. I'm a total microbe nerd. And for those of you who didn't raise your hand, I do hope that some of what I tell you is interesting, and that maybe by the end of this talk you will think that they matter. And if you're into numbers at all, and why would any of you be coming here to the MIT campus? Numbers mean nothing, right? Then microbes should matter a lot to you. It turns out that a recent estimate says that there are 5 × 10⁻30th bacteria on planet Earth. That's 5 nonillion bacteria. That's orders of magnitude more bacteria on Earth than there are predicted to be stars in our universe. So quite an abundance of these bugs, right? And not only are we sort of confronted with these staggering abundances, but we're actually also learning a lot about their diversity. And I'm going to date myself a little bit, but I'll just tell you that this was the view of biodiversity on planet Earth when I was in college. So does anyone recognize this? Whitaker's 5 Kingdoms. Does that ring a bell? Couple heads nodding. Okay, so we're the same age. So basically what Whitaker said was that there was 5 kingdoms of organisms on planet Earth. And you can sort of see, based on this cartoon that I've drawn here, and the sort of circles indicate the relative numbers or the relative diversity that was predicted in each of these kingdoms, that really the plants and the fungi and the animals really dominated, right? Dominated biodiversity. And then here at the bottom, we had some protists, and here at the lowly bottom was the Monera, where all of my beloved bacteria lived. Clearly, you know where I'm going. This is wrong. But it's not any wonder why we may have thought this, because let's face it, how was biodiversity measured? It was measured largely on the things that we could see, right? The characteristics, the traits. Darwin, he used the sizes and the shapes of beaks when he was looking at the finches in the Galapagos to tell him how closely related different organisms were to one another. And he could build these sort of cladistic models that suggested, at least in this case, that finch number 1 and finch number 2 more recently shared a common ancestor with each other than they did to either finch number 3 and 4, based on these physical attributes. Microbes don't have beaks, right? They don't. So here's 2 different bacteria, all right? Who's heard of Bacillus anthracis? Yes, this is a nasty stuff. This is the causative agent of anthrax, right? And who's heard of Bradyrhizobium japonicum? Little less known. This is a really friendly bug, hangs out with plants, does some cool stuff. These 2 organisms, which I would argue you would be very hard-pressed to distinguish if you were looking under a microscope from one another, they are more closely related to each other than we are related to trees. Very, very diverse. And yet, how could you really tell if you were using morphological features? Now, I'll say, we weren't completely in the dark. There are other ways that we could determine different types of microbes, but it's no wonder that we vastly underestimated the amount of diversity in this sort of domain of life. So what's changed? Well, what's changed is that we no longer rely upon things that we can see. We have now switched to using the genomes, the DNA sequence, as a readout of evolution, as a readout of relatedness among organisms. And I'll tell you a little bit more about some of the specifics about this later in my talk. But basically, through this new lens, we can see a completely different version of biodiversity on the planet. And that version, again, it's a cartoon, but it looks something like maybe this. Here we go. Now we've swapped out 5 kingdoms. We've got this 3 domains of life. Here's sort of, you can imagine this is kind of the common ancestor of all life, from which 3 domains that you can see are sort of separated from one another that have emerged over time. And it turns out 2 of these 3 domains are made up entirely of non-nucleated unicellular organisms, including bacteria, these cool bugs called archaea, and then down at the bottom are all of our eukaryotes, so all of our nucleated organisms. And just to kind of orient you to this sort of stick figure, let me show you where all of the plants and the animals and the fungi live. What? They're right there. So basically, I mean, this kind of blows my mind, right? Imagine all of the diversity, the morphological, the functional diversity that exists in these 3 little tiny twigs at the tip of this, what we call tree of life here. And now, look at all of this like deeply rooted branches evolving over many, many eons within these unicellular organisms. Think about how much diversity is there. It's staggering. It really is. And what's cool is that using these new techniques, every time we go into a new environment, we find more and more diversity. These parts of the tree get bushier. We're finding newer things we've never even known existed, while this one stays largely the same. And again, no wonder. We had a pretty good sense of what was going on down here because we could see stuff. Here we had no clue. So you might be able to infer from this that as humans, we evolved in a very microbial-rich world. We came into existence amongst an overabundance of microbes. But it turns out, as we learn about what microbes are existing on our human bodies, it's not just a random sampling of any old bug. It's actually quite specific. Only about 10% of the bacterial diversity that we appreciate today appear to be common residents of the human body. And this is what we call our human microbiome. Let me just see, show of hands. Lisa mentioned the microbiome, and of course it has been in the news a lot. How many of you are familiar with the human microbiome? So you've or heard of it. A few more hands. Awesome, awesome. So though none of you are strangers to the microbiome because you've heard of it, because, well, you all have it. Some of you may not have seen your human microbiome, so I'm going to show you a picture. This is the human microbiome. This is actually a microscopic picture, an image that was taken of someone's supragingival plaque. So this is a scraping of someone's tooth surface up underneath their gums, and now we're looking at it under the microscope. Lots of cool bugs are sort of squished into here. You see these rod-shaped guys and these squiggly guys. And if you look closely, you can see some little spherical guys. It's quite something. And don't judge. Yours would look the same. Okay, so this is a healthy person's microbiome. And what I love about this image is that not only do you see how these microbes sort of closely interact with each other, but they're closely interacting with us. Obviously, they came from this little crevice in someone's mouth, but you can also see features of us in this picture. So here's a red blood cell down here, and then here are features of our immune system. Here's leukocytes. And you can see this by this density that there is a lot of them. In fact, if you consider all of the ecosystems on planet Earth, it turns out our bodies are home to some of the densest communities that there are. And if you just count up the number of these bugs and compare them to the numbers of your cells, you may have heard, who has heard you're more microbial than you are human? Okay, scrap that. It's not true. We revised it. Now the current estimate, you're as microbial as you are human. So there's a little shift. But what hasn't shifted is the fact that in addition to there being many of them, they also encode many, many, many functions. In fact, they encode 100-fold more sort of functions than our own human genome, right? So many more genes. And what's interesting is that though these guys are abundant, though they're very closely associated with us, though they carry all of this metabolic potential, to be very honest with you, even today, we actually know very little about who most of these guys are and what they're doing. We're making some strides, but that's the truth. So, okay, why even care? Why do we even care about our microbes? Well, for one, it turns out, depending on what microbes that you have in your gut, you may or may not be susceptible to toxicity from Tylenol. They actually can interact with chemicals that we consume and make them useful to you, or sometimes make them harmful. So that seems kind of important. They do really cool things. Like sometimes there's this really neat study where they showed how microbes in the guts of people who live in Japan happen to have genes that are sort of specific to the guts of people who live in Japan. And it turns out these genes were taken by Japanese gut microbes from other microbes that lived on the surfaces of the foods they consumed. In this case, it was seaweed. So a lot of nori is being consumed. You have microbes that live naturally in these marine environments on these seaweed plants. They're being ingested. The microbes in the gut say, hey, I'd like to nibble on seaweed too. There's a lot of that around. So they nicked the genes. They incorporated them. Pretty cool, right? Very flexible. Turns out your microbes might tell you who you're going to have sex with, at least if you're a fly. That's as far as I think we've gotten with that work so far. But it is a cool study. So basically, it turns out Drosophila have mating preferences. There are certain flies that they want to mate with and others that they don't. Who knew? But if you treat these flies with antibiotics, thus shifting, right, the complexion of their microbial communities, they lose that preference. Think about that the next time your significant other takes some antibiotics. And then this is one of my favorites. It turns out that there's mounting evidence. It's super cool. A lot of it's in it's mice, but it's still cool. That suggests that the microbes in the guts, at least in this case of mice, can control their behavior. So mice, as you may know, particularly mice that are used in laboratory experiments, tend to be kind of inbred. So their genomes are very similar to one another, and as a consequence, they have very reproducible behaviors. And so this experiment, they used 2 different mouse strains. One was a timid mouse. Apparently you can profile this in the laboratory. And the other was an adventurous mouse. And I'll say, long story short, they swapped the gut bugs in these 2 mouse populations, and guess what happened? Yeah, timid mice became adventurous mice became timid. What? And they were able to track it to the part of the brain, the amygdala. They could see changes in the amygdala that control these kinds of behaviors. This stuff is like chill-worthy, right? It's pretty crazy. Although, Frank, I'll be honest, I think we had many hints of this already in the insect world, of other microbes. So I don't think we should be so surprised. And yet, even when I tell this story, it still freaks me out a little bit. But of course, I think a big focus of why we care about our microbes comes from work. You can come in. Okay. You can come here. With this concept that shifts in our microbes, abnormal microbiomes, or you'll hear me maybe use the word dysbiotic microbiomes, are associated with different diseases, right? And so if we think about the mouse experiment, where you can shift the behaviors of mice by shifting their microbes, could it be the case that if microbes are to be sort of implicated and responsible for some of these disorders, that simply by modulating the bugs that live in and on us, perhaps we can overcome these? So that's the brass ring. That's what everyone, I think, is very excited about. But I'll tell you, as these kinds of results were coming online, there was a big question mark, and that was, well, okay, we say that shifts might be important for disease, and that maybe by reverting those shifts back to something normal might be helpful, but what is normal? What does that even mean, right? And this is where the NIH, National Institutes of Health, sort of stepped in about 10 years ago now to really lead the charge to define what it meant to be a normal microbiome. So in many respects, it was like a second human genome project, right? But instead of sequencing our genomes, we're trying to understand and characterize the genomes of all of the bugs that live in and on us in healthy people. So I'll just mention that Broad was one of the 4 sequencing centers that was involved in this many-year, obviously multi-collaborative project. So I'm going to be speaking about just a little slice of it and on behalf of a lot of people. Actually, I joined the Broad when I joined the Broad 7 years ago. This is the project that I worked on. And it was pretty fun. Okay, so if you want to create this foundational information about healthy microbiomes, you first got to get some healthy humans with healthy parts. And although I wasn't there, I've been told that at the beginning of this project, there was a lot of discussions with ethicists and clinicians and academics, and you'll find out later, a lot of dentists apparently, who all sat together and sort of thrashed on what it meant to be healthy, a healthy individual. And they came to some apparently very strict criteria for this, including, for some reason in this study, you needed to be between 18 and 40 years old. You needed to have a certain BMI, and you had to be healthy, clinically healthy, at all of 18 different body habitats if you were a female, and 15 body habitats if you're a male. And of course, you also had to agree to be sampled at each of these body sites once and sometimes more than once. And that's what happened. Most of these are sort of swabs, not invasive kind of stuff, because healthy people, I would guess, are not too excited about getting more in deeply sampled than that. For the gastrointestinal tract, if you're wondering, this was poop. So that was our proxy for the GI tract. And over time, we enrolled 300 healthy individuals, collecting over 11,000 different samples, many of which came from time points, multiple time points along the way from the same individuals. Okay, so what do we do with all these samples? Right? So you've got all these swabs and these scrapes, and you want to characterize who's there and how many. And that's exactly what we wanted to do. It's sort of like a census, right? You knock on someone's door, you ask, all right, how many of you were in there? You know, what's your ages, what's your races, so we can sort of categorize you and bin you. And that's exactly what we wanted to do with each of these samples. A couple different ways to do it. One would be the old-fashioned way. Take these samples, you grow them on a bunch of different media in the laboratory, and then you characterize painstakingly them one by one by one. We didn't do that. One, because that would take forever. And two, because frankly, we actually don't know how a lot of these things grow or how to make a lot of these things grow outside of the body. So we would have been completely sort of blind to a large fraction. So what we needed was a way to do this census that ended up being what we call a culture-independent method that also enabled itself to high throughput. I mean, 11,000 samples is not a small number. And so, of course, we wanted to leverage the power of sequencing. And so we used sequencing technology to generate these bacterial signatures that then could tell us who and how many were in each of these samples. Okay, so to give you a little background on what this is really all about, I want to introduce you to a ribosome. Who's heard of a ribosome? Awesome, awesome. Okay, so for those of you, I'm impressed. So this is part, central dogma. You have DNA that gets replicated to DNA, and then you get transcribed to mRNA, these messenger RNA, that then go on to be translated into protein. Well, that translation piece going from mRNA to protein is dictated partly by this molecule here, this ribosome. If you don't have a ribosome, if you're not making protein, you're dead. It turns out the components, it's a multi-component structure, this macromolecule. It turns out that the pieces of this, we believe, were in the common ancestor of all life, okay? So it was derived from the common ancestor of all life, and because of the essential function of this macromolecule, and because all these things have to work together in such a way, you can't really tweak it too much. So as organisms have evolved, only some changes have occurred. And that's great. That's cool. Because what happens, oh, it doesn't come through so great, is we can see, I'm just giving you a little slice of a sequence alignment of a gene that encodes for one part of this ribosome, right? Where you can see, hopefully, through some of the shading, regions of the sequence going from humans to yeast to corn to bacteria to these archaea that are highly, 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 highly conserved. This doesn't happen in other genes in our genomes, right? This is not the way it works. But for this gene, it works really nicely. So you get these highly conserved regions, and then you get these regions that are sort of in between that are variable. And so this lends itself to us being able to develop sort of molecular methods that allow us to target these variable regions using these highly conserved sites, right? So we know how to like pull it out and then get this sort of variable region that we can then use as a sort of clock, sort of an evolutionary clock, by simply looking at the numbers of changes in these variable regions as well as the types. We can start to infer something about how closely related two things are to one another. So in this example, again, I'm showing you sort of this tree. That, again, you'll probably hear me use that word quite a bit, where we've got E. coli and this, in this case, I forget, this is a cyanobacteria, I think, clearly more recently shared a common ancestor than either did with either a yeast or a human. So this is the basis of it. So we can use these tags to tell us something about the identity and relatedness among these organisms. So let me pause for a second and see if that makes sense. Is everybody with me? I should probably ask in reverse. Who's not with me? Okay, you know, even if you're not going to be lost for very long, I promise. Okay, so what we can do, again, not wanting to get too much in the details, but wanting to help you understand how it is we profile these communities, is we can take one of these complex samples, so these, say, a nasal swab, get its DNA, use our molecular approach to pull out all of these variable regions from each of the genomes of all of the organisms that live in this community, and then sequence it. And then from that sequence, we can give each of these sequences a name, right? We can characterize it. And here I'm just indicating different flavors, different names of these variable regions using color. So we have a bunch of blue, we have some green, we have some red. And oftentimes, you know, we'll sort of just reduce this to numbers. We'll count them up. We'll figure out the frequency. A lot of people like to look at this information as a heat map, right? And that, so that if you look at multiple samples, you can begin to get a sense of the relationship based on that community profile. Okay, so that's what I'm showing you here. We've now looked at 3 different samples, and based on the frequencies of these different tags that we've solved, we can see, for instance, that sample A and sample C look more like each other than either do to sample B. It's actually kind of simple. So with that in mind, I'm going to take you to some real data. So here are the data I'm going to show you come from 242 of our healthy volunteers who were sampled across, in total, 18 different body habitats, over 5,000 samples, and here's some details about how deeply we sampled each one. So in theory, we're getting a readout of 6,000 or so members from each of these different samples. So what do the data look like? All right, I tricked you. We're not going to look at a heat map. we're going to look at this instead. So all you need to know to understand this graph, so I've put all of the data points on here, and each one of these circles represents a different community profile. Okay, so that's one. The closer 2 circles are to one another, the more closely related or similar their community profiles are to one another. And then the other piece of information that's probably obvious is that I've now color-coded and given names to where those different communities came from across these 200 individuals. So stepping back, I think one of the take-home messages that I hope that you can get right away is that the site from which the samples were taken tends to be the major driver of how similar they are to one another, right? So in other words, the bugs in my nose look more like the bugs in your nose than the bugs in my nose look like the bugs in my mouth. And that kind of makes sense, right? I mean, if you were to look at plants in a tropical environment and compare them to plants in a dry environment, they're probably different. And I think it's the same principle, sort of the ecology of the sites from which these communities reside sort of dictate who is there and obviously what they're doing. So that was great, and I'll just say that this work sort of supported some of the work that had been done previous to it, but I think the power of this study with so many subjects and so many samples was that we really started to get this sense, the boundaries of what normal really was, and that's what we were after. And though you can see in this view that, let's focus on the gastrointestinal tract, normal gastrointestinal tracts look a lot like each other based on this view, right? Especially when you compare them, to skin or to something in your mouth. But when you really dive deeper, it's actually a bit more complicated. It turns out healthy can vary quite a lot. So now what I'm showing you is for 200 of the individual volunteers. I'm now breaking apart the different components of the communities that we solved. And I'm showing you on the y-axis the relative abundance of those members within each of these 200 samples. And so I hope what you could appreciate is that there's a wide, obviously we've sorted it based on this one sort of dominant member, this Bacteroides member. And you can see that some people are almost exclusively Bacteroides, and some people have very little. And instead they have a bunch of this other stuff. So there's a lot of diversity within normal. So it's not just as simple as your bugs look just like my bug. No, yes, we have the similarity that makes us, it makes it so you would know where her bugs came from in a sample versus mine, but you wouldn't necessarily know that it had this much or whatever, right? And it turns out that is the pattern that we see across all of the body sites that we looked at. It's not just the gut. It's different sites in the mouth. It's in the vagina. It's a little bit different in the nose. There seems to be like 2 major species that kind of oscillate. But overall, healthy varies quite a lot. So that's one major message. All right, but the real fun of these data is when you start comparing them to other data sets, right? The power is really in the comparisons. And so I want to take you through one example that I think is kind of cool. Well, I hope you think it's cool. And that is an example where comparing these patterns started to give us some insights into where our microbes came from. So, the dogma is that each of us is born sterile into this world. Actually, that is being debated a little bit, I'll just tell you. So I think the jury's out whether that's 100% true. But that's the dogma, that we're born sterile, and yet we all get colonized with bugs that end up leading to these very consistent patterns. So what's up with that? Where do these bugs even originate? So we can start to break that down. So here I'm just going to start using a similar kind of construction. I'm going to start building on a couple different data sets to walk you through it. So here's a data set of community profiles coming from an elder population, from their guts, that when we compare to the cohort of people who are enrolled in the Human Microbiome Project, that again, this is gut communities, we can see there's a nice overlap, suggesting that even though in our study 18 to 40 was sort of the cutoff, you can be older than that. And actually, there's not a big shift in what a gut microbiome community really looks like. We can see that there was nothing special about our study. Healthy humans from other studies look similar. That's great. But when we took data from studies that looked at the communities within infants' guts, it looked different. It looked more like this. So certainly, for the large part, they weren't sort of overlapping with healthy adult guts communities, but they had sort of these 2 tails. And what we started to see very clearly when we overlaid data from other body sites was that these 2 tails appeared to overlap one of 2 body habitats, either communities that come from the vagina of healthy adults or communities that come from the skin of healthy adults. And so why would that be? Any guesses? That's right. So what she said is that, And she's right. So in this case, babies whose gut profiles looked more like vaginal profiles were born through a vaginal route. Babies whose gut bugs looked more like healthy human skin were born through C-section. Exactly. So it really does indicate your first exposure sort of dictate what bugs sort of begin to inhabit and take hold into your habitat. But what's cool, and actually we just recently began to learn about this, is, I mean, let's ignore this. It appears that everybody, regardless, converges here. So what's up with that? How long does that take? Turns out it takes about 2 years. So regardless of which route of delivery you come from, it takes about 2 years, and you start to converge on this adult conformation. Kind of cool. Okay, but we can do similar things looking at health and disease. So here I'm showing you data from an older study now, where basically I've taken away color. So this is a graph from a paper where they've plotted communities coming from healthy individuals compared to communities coming from individuals with ulcerative colitis and individuals with Crohn's disease. And in this case, and in a lot of cases like this, these investigators could see a separation, suggesting that there was something special about the communities living within the guts of people with these disorders as compared to their healthy controls. And so I can tell you that our field, the microbiome field, is very engaged in these kinds of patterns. And the question of what's driving what, right? It's a bit of a chicken and egg question. Is it that this different configuration of gut microbes in people with ulcerative colitis is what's driving ulcerative colitis? Or is it that people with ulcerative colitis have a different sort of gut ecology that selects for different configurations of bugs? And this is really where, like I said, there's a lot of effort that is going here to try to sort this out. And I think we will see more and more and more studies related to this in the future. But what I want to do is I just want to say that among all of the kinds of disorders and diseases that are being examined in this way and that have started to show these kinds of patterns, I think it's disorders like UC and Crohn's and autoimmune inflammatory disorders that seem to really scream for a microbial component. So again, much more research needs to be done. Actually, a lot of really beautiful work is being done here on this very question, but I think we still have a ways to go. But the reason I say that is because you're probably well aware that these rise, as I'm sure you're aware of, in the incidence of these autoimmune disorders doesn't seem to be explainable by human genetics alone. We know human genetics have some role in some of these disorders, but the increases are dramatic, and they cannot be explained by our evolution, right? We can see in only a matter of a few decades, we see these increases in asthma and diabetes and multiple sclerosis and Crohn's disease that, interestingly, are anti-correlated with certain infectious diseases, right? Like measles and mumps and TB and hepatitis. This sort of screams of an exposure, but maybe it's really lack of exposure. And while it's formally possible that these actual infectious diseases may have a role, it's possible that it's also really giving us sort of an indication that it's sort of a proxy for our overall exposure to microbes, right? And I think supporting this is this graphic, where we can see when we look at the incidence of these autoimmune disorders around the globe, it's largely the westernized parts of the globe that have the highest frequency. So the red and the orange are the places where we see more of these kinds of disorders. And you're probably already there, right? These are also the places, frankly, where we are most hostile to our microbes, right? We don't live with our animals. We purify our water. We disinfect a lot. We really create a lot of boundaries between ourselves and the bugs that we evolved with. So who's heard of the hygiene hypothesis? Right, so this is sort of the basis of that. And there's a lot of evidence. Again, I think there's more work that needs to be done, but there's certainly a lot of evidence that are mounting to suggest that, this may be true. In fact, that maybe the lack of exposure to certain bugs early in life, or the removal of certain bugs via, say, antibiotics early in life, can possibly set the stage for sort of long-term immunological consequences. Again, lots of work needs to be done, but these are the trends that we see, and I think that there's going to be a lot of interesting work in this space coming along. Okay, you know what? I'm going to take a pause, and I'm going to see if people have any questions. Right here. So the question is about, so you're interested to hear more about the diversity.
Right, so basically we have a system.
Yep.
Functioning of that system in the human context all the time actually depends on how parts interact.
Yes.
So I was just wondering if you could talk to that. And then the other thing is, there have been studies that look at the human between genetic,
Yeah.
well, dynamic and microbiome.
Ooh, okay. No, they're both great questions. So one is about community dynamics and sort of how the microbes interact with each other, presumably, right? How they sort of configure.
We're dealing with systems.
Yeah, yeah. 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 So that is another really hot area in the field, trying to make sense of the communities as a thing, as a system, as you put it, right? And I'll say, what's kind of cool, it's not directly answering your point, because I think there's still a lot of work that needs to be done. How do these things interact with each other? How do they interact with us? What's the sort of feedback loops that are sort of set up? And how do you measure that interaction? Great questions. And so there's a few ways that one can begin to tackle it. I'm trying to think of what level we want to go to. But I think that's the systems biology piece of this is like a next frontier. And people are starting to crack away at this. So we can look not just at 16S genes, right? So far I've only shown you data looking at a single gene. But of course, each of these strains, each of these organisms carries a whole compendium of genes. And all these genes have different functions. So how do they function even within this one organism? But of course, the question beyond that is how do they function within this superorganism, if you will, that is the community. And so we can look at the genes and look at the prevalence of those genes, what the genes are, can we predict what they do? We can take it to the next level. We can look at how those genes are being expressed, right? In certain conditions, in certain environments. We can actually take samples directly from your nose and tell you about who's transcribing what. But again, these are readouts, but I think the more complex piece is how to put all those readouts together to make full sense of the system. And again, there are some algorithms that are being developed to try to sort of tackle this. Are there anti-correlations between different pathways? Are there correlations between different bugs or anti-correlations between different bugs? And how does that relate to the host? These are super important questions, but they're actually tough. They're pretty tough, but I think it's where the field is going as well. And then you asked about the host, right? Have people started to look at how communities relate to, say, host genetics or host epigenetics? And again, I think, yes, people are looking at this, trying to determine whether or not certain genetic profiles of an individual could dictate whether they are a high Bacteroides, for instance, or a low Bacteroides. I'll tell you this, out of all the metadata that were collected as part of the Human Microbiome Project, the only piece of metadata that was strongly associated with anything having to do with the 16S communities was race. That was it. That was not, so that screams genetics, right? There's something different, functionally different. That's sort of, well, that's one explanation is genetics. It could also be exposure. That's the other thing. Maybe diets are different. You know, we don't actually know. So I think again, this is some unsolved questions, but territory that people are really hot about trying to answer. I probably didn't answer all your questions, but they were great ones. So maybe take one more, and then we'll move on right here.
So are there any specific before and after antibiotic studies, you know, like on humans or animals or whatever?
Yeah. Okay, I love that question, and it's sort of like a nice segue for the next section. So before and after of the microbiome, so I won't touch on that, but there have been, and antibiotics, as you might imagine, does have a big impact on the microbes. You know, a lot of focus has been on the gut, so it has a big impact. You do see rebound, so it's interesting. There hasn't been that many really what I would call very nice longitudinal studies in this regard, but I think, if I'm remembering correctly, there might be people in the crowd who know this literature better than I do. So please feel free to speak up. But I believe that overall, you do see a resilience. So though you may hit it with antibiotics, it changes, but oftentimes you see some rebound of the community. I don't think it's perfect, though. There can be long-lasting impacts. I've also seen some neat data on food poisoning. That there's one study where someone sort of looked at their gut microbiome every single day for over a year, and they happened to get salmonella poisoning, and they decided not to take antibiotics because they were excited to see the results. Crazy. It's kind of crazy, I know. But what they found was actually their microbiome never came back, at least in the study period. So it major crash, like they lost a lot of diversity. You saw this big pathogen bloom of salmonella in their guts. And then although the salmonella went away over time, it never came back to what it was. It was different. So I think these perturbation studies are super cool, and we probably need a lot more of them. But antibiotics are very important. They're very important, so I want to talk about them. Obviously, antibiotics, what's my time? 5 minutes. Okay, 10 minutes, we got this. All right. So antibiotics. So obviously, antibiotics is something that we should be very thankful for, right? I mean, this has saved millions of lives. But as you're probably well aware, there's been sort of a rebound effect. Now there's the rise up of pathogens that are now resisting our ability to treat them with the antibiotics that we have. So who here has heard of like superbugs? Yeah, it's a big deal. Actually, just yesterday, there was something on CNN about how in Rio they found superbugs in the water, right? Who saw that? Yeah. So this is a big deal. I mean, so CDC put out a threat report a couple years ago that said, you know, there was 2 million illnesses related to AMR disease, you know, 23,000 deaths, which doesn't sound like that much, although obviously it's important. But I'll just tell you, a newer study that I won't show you data for suggests that by 2050, so not so long from now, there will be 10 million deaths each year attributable to antimicrobial resistant related illnesses. That's like 2 million more deaths than the number of people who die from cancer each year today. That's a lot of people dying. And there's many reasons why I think we see this rise in resistance. That could be the subject of an entirely different talk. But I think one of the reasons that we have to consider is the way that we treat these precious resources, right? We don't do ourselves a lot of favors. For one, we use a lot of antibiotics in this country. You know, certainly in clinical use, 7.7 million pounds of antibiotics are brought into clinical settings each year, which does sound like a lot. And while I think that doctors are using them well in many cases, I mean, how many of you get antibiotics and you don't even know if you have a bacterial infection, or at least been offered them? You know, it happens a lot, and that's not great. But I think what sort of dwarfs this is the amount of antibiotics that we use in agricultural settings. You mentioned it. Yeah, your finger's on the pulse here. So it turns out it's like 3 times more. And I'll tell you, this is not to treat sick chickens. This is largely being used at subtherapeutic levels to quicken the pace at which animals build protein, right? We don't even understand how that works, but that's largely what it's being used for. And I'll just tell you, if you're an antibiotic and if you're a bacterium and you want to evolve resistance to antibiotics, you want subtherapeutic chronic doses of antibiotics over time. So this is like the perfect breeding ground for this problem. And I'll tell you more than just sort of hand waving. I'll tell you a story about it if you don't believe me. So this is actually some other work from my group, more recent work from my group, in collaboration with other academic groups in the area. So once upon a time, there was this harmless member of the microbiome called Enterococcus. Who's heard of Enterococcus? All right, more than Brady Rhizobium, that's good. That emerged as an epidemic multi-drug-resistant clinical menace. And this is not the menace, this is my, although he's a lot of fun, I have to say. This is my buddy Francois Lebreton, who I worked with on this project. And so what Francois did is he really wanted to understand. He came to us, and he was very passionate about the enterococcal problem, and he said, I want to understand how this has happened. How did we go from having an organism that has been with us since we existed? I mean, this bug was in the microbiome of dinosaurs. This is some old stuff. How did it go from being such a commensal happy bug with us to being such a problem? It's one of the second leading causes of hospital infections, of bloodstream infections. This kills a lot of people. And so what he did, in a nutshell, is he selected a diversity of these isolates within Enterococcus, in this case faecium. So that's the name of the species. So representing sort of the diversity, that's kind of what this shows. We don't need to worry about this image so much. But they were not only diverse in terms of what we understood about their genetics, but they were also diverse in terms of the time in which they had been isolated, so over a 50-year period, as well as from where they came. So humans and animals and hospitalized humans, you name it. So we took these isolates that Francois selected. We brought them into the lab. So I'll just show you a little bit about how we sort of do this work. Grew them up in the lab, isolated DNA, did some QC stuff, who cares? Bottom line is we see, okay, it is important. You should QC. But bottom line is we were able to take the DNA from these organisms. Instead of just sequencing a single gene, we could sequence all of the genes, right? We could actually recreate the genome of these organisms in sequence space. And using the data from all of these strains, we could start to ask a lot of interesting questions, like how do these strains relate to one another? So I'm going to show you some data here, but I'm going to hold your hand a little bit. So what you're seeing here is what we call a phylogenetic tree. It's based on sequences that are common among all of the organisms that we sequence and analyze. And the way you read it is as follows. So basically, each tip represents a different isolate that we sequenced. And the closer that 2 isolates are to one another via these horizontal bars indicates their closeness in sort of genetic space. So these guys are much more closely related to each other than they are to, right, guys that are down here. Does that make sense? And so what we could see when we looked at this tree topology was that there were some very distinct and robust clusters of organisms. So they clustered very clearly. And when we looked to see where these organisms were derived, and also we could go to other databases where we understood more about the propensity of these different types of isolates living in different environments, we saw that these clusters actually corresponded beautifully to habitat. So it turned out that organisms that sort of clustered up here tended to live in the guts of healthy individuals. We call them commensals instead of pathogens, that's some verbiage. The bugs down here tended to live in healthy animals. So these were isolated from animals, domesticated and otherwise. And then the organisms that tended to cluster here were those that were found in hospitalized individuals. So these were sort of our hospital-adapted group of strains. So there's very strong structure. And of course, we had to ask ourselves, well, well, why? What has led to this topology? What has led to this sort of evolution? And why is actually a really hard question to answer using genomics. That's a hard question to answer almost any time. But what we could ask with these data is when. So remember, just like with the 16S sequences, this is sort of this tree topology and the sequence data and comparing across the different sequences, it serves as sort of a clock. So we could actually date timing of when some of these sort of events that led one group to be different from another, when those actually occurred. And what we found was when we asked, okay, so when did strains that were living in healthy humans sort of begin to diverge from strains that lived in animals? And what we calculated was that this happened about 3,000 years ago, which just so happens to correspond roughly to the Iron Age of human existence. So this is when humans stopped living with their animals. They moved from more rural settings into more urban settings. And frankly, what we think is stopped sharing their bugs back and forth. And so humans and animals sort of became their own little islands. So they took their little ephysium and they walked away, and that ephysium evolved with them and became a distinct thing, a distinct population. Okay, it's harder to see, but there's this very robust sort of cluster down here that we could also, actually here's, oh hell, I can't even tell you where the node is, but it's real. I can't show it to you, but I know it's there. So when did this occur? When did this split between animal commensal and hospital adapted strains occur? Any guesses in years?
50.
Pretty good, pretty good, pretty good. We estimate about 70 years ago, which just so happens to be, and there's error bars and all of this, so it could be 50. It could be, which just so happens to be around the time that antibiotics were introduced, right? And so what we think, again, is that these populations began to respond to the presence of a new environment. And we think that new environment was life in animals that are being treated now with antibiotics that set the stage for them to move into these new clinical settings and in hospitalized patients. So let me go quickly. Obviously, in addition to looking at relatedness, we can also look at what genes are being sort of held within these different strains and compare them to one another. And one of the clear messages was that strains from these hospital-adapted isolates, as well as the animal commensals, tended to carry a lot more resistance genes. So each row is a different gene that we know confers resistance to antibiotics. Red, it says it's present. White says it's not present. And you can see in commensal strains, there's hardly any antibiotic resistance. But in these other populations, there's a lot. So that helps to explain why they may be there. But what's even cooler, because that's kind of expected, but what was even cooler to me was when we started to look at pathways that dictate the kinds of things that you eat, so the kinds of sugars that you eat. And what we saw, again, not too surprisingly, was that strains that lived in healthy humans tended to have these sugar eating capabilities that were quite distinct. So they had different profiles from those that lived in animals. Kind of makes sense. The diets of humans and animals differ, so that made some sense. But what was really cool was that in these hospital-adapted strains, there seemed to be this other system, this other, we call it PTS system, I might say that, carbohydrate utilization system, that was almost solely enriched within the hospital-adapted population. And it turns out that we predict that this utilization pathway enables these strains now to eat something called mannose, a sugar called mannose. Turns out mannose is a sugar that's highly abundant in mucins, which are, of course, mucins are what sort of decorate your gastrointestinal tracts. So what we think the model is that commensal Enterococcus faecium lives in an environment in a healthy individual where that individual is likely eating food, normal food, so they're getting exposure to food. They're around other bugs, so which may be supplying other kinds of nutrient sources and perhaps food. But when you start to get into these hospital-adapted strains, now they're red because we know they're resistant to a lot of antibiotics. So a hospitalized patient comes in, chances are they're being bombarded with antibiotics. These guys survive because they're resistant. The commensal population goes away because it can't survive, right? Chances are the patient may not be getting sort of oral nutrition. Maybe they're on IV, so there's no real food around. And so what we think happened was kind of like the seaweed example, right? Where you had availability of something that the bugs couldn't use, but they said, that would be a good idea. I'd like to use that. I'm going to take those genes. I'm going to try that on for size. And if it works, it sticks around. So what we think is that these guys tried on a new sugar utilization pathway on for size for mucins that now enables them, in the absence of other nutrition, to begin nibbling on the guts of patients and surviving. Pretty crazy. All right, that's it. That's it. That's it. OK, model, genes, environment, disease. We got to think about the microbes. Take-homes, they are us, right? They influence us in ways we're only beginning to understand. We need to respect our microbes. We're squandering precious resources. And frankly, this is some scary space we're getting into. And so I'll just say it's causing us to need a great deal of more research and more strategies for dealing with it. So with that, I want to thank you, and I want to thank some folks. Bruce, my boss, who's fantastic. Abby, who did a lot of work on the Enterococcus project. Here's our Enterococcus collaborators, and a bunch of other people who are amazing to work with here. Thank you for your attention.